नमस्कार दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing another topic that is drug-induced liver injury, DILI. So let's see what is the question. The question is a clinical assessment of drug-induced liver injury using Hayes law. So something new term that we will be learning in this video is about Hayes law. Consider what test results. So basically we'll understand from this video about drug-induced liver injury. Also we'll learn about what is Hayes law. So option A we have greater than threefold increases in serum ALT and greater than twofold increase in total bilirubin. Option B, we have threefold increases in ALT, AST, and ALP without increased total bilirubin. Option C, we have a greater than threefold increase in ALT, AST, and greater than twofold increase in total cholesterol. Option D, we have threefold increases in ALT, AST, and ALP with twofold increases in total bilirubin. The right answer will be in the green text, that is option A, threefold increases in serum ALT and twofold increase in total bilirubin. So serum ALT threefold increase, total bilirubin twofold increase. So let us see what is the explanation and the reference which is given by the American Board of Toxicology. Hayes law is a useful as a prognostic indicator in drug-induced liver injury. So, particularly for hepatic injury, this particular law is applicable. What does this law? It combines serum ALT, that is threefold serum ALT, and three times ULN. What is ULN? That we will understand. And serum total bilirubin, what is two times of ULN in making a prediction of outcome. Why do we need Hayes law? Why there is a threefold, twofold, and why there is a combination? All these things we'll understand in the next slide. So in addition, also you read the American Board of Toxicology has given the reference Casret and Dole 9th edition, page number 732 and 733. Okay, let's see what we can learn from this one. And additionally, you also make a list of drug-induced liver injury. So what are the drugs? What are the different classes of the drug which are causing liver injury? And particularly similarly like kidney toxicity, you identify which part of the liver, what a particular lobule or cells, and what type of injury these particular drugs are causing, whether it is causing cholestasis like that, cirrhosis. You also try to read the and remember the particular cell type of damage, liver damage it is causing. Okay, so let's directly read page number Casret and Down 9th edition, page number 732 and 733. In this explanation is also given, and also they have given a table in 13.3. This particular ratios, R ratios they have given. So let us understand what is first this one, R value, as well as what is this Hayes law. Because values for enzyme activities in serum and plasma from normal healthy people can vary across clinical chemistry laboratories that perform the measurements. For example, we have one clinical laboratory 1 and another clinical laboratory 2. Even if they are performing the same ALT AST, there is a difference in the laboratory results with the same patient or same parameter. I'm saying same assay. So to overcome those differences, laboratory differences, increases in the enzyme activities are typically expressed as ratio normalized to the upper limit of normal. That is what it is called ULN. If you see in the previous slide, no, ULN was measured, no, two times of ULN. So what is this ULN is nothing but it is the upper limit of normal for the particular laboratory in which the measurement was performed. So we have for a certain laboratory, a certain ULN value will be there. In humans, serum ALT activity that is 3 to 5 times or more above the ULN suggests a liver injury. So whenever there is a ULN value more than 3 to 5 times more, then serum ALT activity is more than 3 to 5 times more the above whatever the ULN suggested in that particular laboratory. In that case, it is considered as liver injury. Next, clinical drug-induced liver injury is often classified as hepatocellular, cholestatic, and mixed, that is both hepatocellular and cholestatic, depending on the clinical chemistry results. As I said, no, we have to identify what drug and what type of within the liver also what type of injury it is causing. For this classification, the so-called R value. What is this R value? R value is calculated from serum enzyme activities, where R is nothing but ALT divided by ALT upper limit of normal, and ALP divided by ALP of upper limit normal. Okay, first you are directly taking ALT, in the denominator you are taking ALT of upper limit normal. So if R is greater than or equal to 5, then it is the injury is considered to be hepatocellular. See here, you can see in the table 13.3 they have given. If the R value is less than or equal to 2, this one, then it is called the type of liver damage it is causing is cholestatic. And if the R value is greater than 2, less than 2 and less than 5, then it is mixed type of hepatic injury. So this provides a conceptual framework for the diagnosis of the liver injury. So please try to remember this table 3.3, understand what is ULN, what is Hayes law, what is the R value. Okay, useful in predicting clinical prognosis in patients with liver injury is Hayes law. So Hayes law is based on the observation. This is a particular person, Dr. Hyman Zimmerman, that the combination of serum ALT and greater than 3 times of ULN and total bilirubin 2 times of ULN in patients with drug-induced liver injury portends a poor outcome, that is a risk of 10% mortality. So first we have to understand what is R law, R value. Then you have to apply the high law is a combination of two things. One is serum ALT this value and total bilirubin. So this is what we had the form. This is R value. So let's see what is Hayes law. Hayes law, the better understand, is a rule of thumb that predicts a high risk of severe drug induced liver injury when a patient shows hepatocellular injury, damage to the liver cells, either with the jaundice, that is yellowing of the skin as well as the eyes, after taking a certain drug. It is a clinical tool based on observations by Hyman Zimmerman and is used to identify drugs that may be particularly liver toxic, with the criteria being an elevation of the liver enzymes. What are those enzymes? ALT, AST, and a rise in the total bilirubin. So these are the three biomarkers, clinical biomarkers. Okay. So key criteria is elevated liver enzymes, elevated bilirubin, exclusion of the other causes. What are the elevated liver enzymes? Either ALT or AST is at least 3 times of the upper limit of the normal. And coming to elevated bilirubin, total bilirubin is at least 2 times of the upper limit of the normal. And exclusion of other causes, there must be no other reason for the liver injury. Exclusively, these are the two parameters should be there, and as well as the drug which is the chemical which is causing this injury, such as a viral infection or pre-existing liver diseases. Okay, these two should be not be whenever you are using Hayes law, these other two factors should not be involved in that one. So this is the typical pictorial representation you can see. Hayes criteria includes AST or ALT, which is greater than ULN 3 times, and total bilirubin, see, 2 times greater than or equal to your normal or increased ALP, that is equal to hepatic cellular injury. This you have to remember in your this thing for your exam. So in addition to this concept, we also see what is the importance of this Hayes law as well as this R value. It is used for risk assessment. It is also requirement by the FDA regulatory agency. It is used as a predictive power, and it is also used for drug development. So what we have in the case of risk assessment, this Hayes laws will help identify drugs that have potential to cause fatal liver failure. Even though we know that liver can regenerate, still there are patients who undergo fatal liver failure. To identify that, we can use it. This you can Hayes law can be used as a risk assessment tool. Second one is the US FDA has a modified version of Hayes law to assess drug safety during initial clinical trials only. We don't want any patient who are participated in the trial to undergo any fatal liver failure. So in order to contain that also, FDA, the modified version of this Hayes law is there, which can be used at the beginning of the clinical trials only. Third one is it is used as a predictive power. While it has limitations, every law, every method essay will have some limitations. The law is specific for predicting severe reactions and has been confirmed by prospective studies. So maybe whenever there is a severe kind of liver damage, that time only this particular can be used as a tool. Drug development, it is a critical tool, Hayes law, for pharmacovigilance to identify new compounds that may be causing liver injury. So these are the different applications, the importance of this Hayes law. So in addition, in this video, we'll also learn about Another important thing, that is table 13.2. So I have taken directly from the lesson, page number 726. You read the types of liver responses to xenobiotic agents. This is very important. Within the liver also, based on which region, which cells are getting damaged, there is a response. So there will be hepatocyte death, fatty liver, that is steatosis, canalicular cholestasis, bile duct damage, sinusoidal endothelial damage, inflammation, liver regeneration, fibrosis and cirrhosis, and liver cancers. So what are the representative and toxic agents? So in the exam they will ask you about they will give you examples only. You need to identify what kind of damage it is causing. So here, acetaminophen, allyl alcohol, metals may copper, dimethyl formamide solvent, ethanol is also solvent. So these organic solvents, they are causing hepatocyte death, complete death. Okay. Next one, amiodarone, CCl4, carbon tetrachloride, which is also an example for your synergistic effect. Ethanol solvent, fluorouracil, tamoxifen, which is a breast cancer estrogen antagonist against this one. Valproic acid, anti-epileptic drug. So these are the examples what they're causing. Fatty liver cirrhosis. So important thing you have to remember, ethanol, alcohol is causing fatty liver, carbon tetrachloride is also causing fatty liver. Then next, coming to canalicular cholestasis, what are the drugs which are causing? Chlorpromazine, cyclosporin A, solvent 1,1-dichloroethylene, estrogens, metal against it. Manganese is causing canalicular cholestasis and phalloidine. Okay, phalloidine. Coming to bile duct damage, alpha naphthyl isothiocyanate, amoxicillin, methylene dienine, and sporidesmin. So these are the chemicals or drugs what you can say which are causing bile duct damage. Coming to sinusoidal endothelial damage, we have example cyclophosphamide, anti-cancer nitrogen mustard drug, microcystin, pyroxolidine alkaloid. So these are the examples which are causing sinusoidal. In the liver, sinusoidal portal will be there, no, that one endothelial damage it is causing. In my first picture I given the different regions of the liver. There you can see inflammation of the liver is caused by a bacterial endotoxin as well as alcohol. Now you see alcohol is causing hepatocytic death, alcohol is causing fatty liver steatosis. So alcohol is affecting all the major parts of the liver. Okay, and it is also causing inflammation. Coming to liver regeneration, acetaminophen, ethanol, estrogen, again alcohol is involved. And if you see acetaminophen, it is causing hepatocyte death, it is also impairing the liver regeneration. Okay, because of which one is that there is permanent damage, irreversible damage. Fibrosis and cirrhosis, again alcohol, carbon tetrachloride, thioacetamide, vitamin A. So much of the following vitamin, then you have to remember vitamin A causes fibrosis and cirrhosis of liver. Vinyl chloride, which is used in rubber industry, a major part of the plastic material also, vinyl chloride. And maybe it is used as a solvent also. Coming to liver cancers, we have aflatoxins, androgen, arsenic. So metal which is causing liver cancer is arsenic, thorium dioxide, and vinyl chloride. So vinyl chloride is causing fibrosis and cirrhosis as well as it is causing liver cancer. Metals may arsenic, liver cancer. Then we have what other metals we have studied here? Copper. Copper is causing hepatocyte death. And there was something else. Manganese is causing canalicular cholestasis. So like that you have to remember. Solvents, what part of the damage, metals, whatever. So which class of drugs? Whether see most of the time we have come across tamoxifen. Anti-cancer drugs are cyclophosphamide is coming here. See, then we have other type of anti-cancer drugs are also there. So remember in that way that will be helpful for your exam. Okay. So hope from this video you were able to understand about liver toxicity. Majorly we have learnt about R value, High's law, the ratio ALT AST, how once you perform the parameters, how to use the data to make it into a working model, the tool, how it is useful during initial clinical trials as a tool in the case of risk assessment also. And we have listed out some of the drugs also which are causing the different types of liver injury. So hope you learned a lot from this particular topic. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. Thank you so much. All the best for your exam.